Hello, this is Adam Paulson welcoming you to this bite-sized bio webinar. Today's presentation is titled The 12 R's Framework, Enabling High-Quality Humane Research Involving Animals, and is being presented by Dr. Dave Lewis from the University of Leeds. Dave Lewis is a senior lecturer in pharmacology and ethics at the University of Leeds. His interests lie in enabling high-quality humane science involving research animals. He collaborates with colleagues in the global North and South to create professional educational opportunities for all stakeholders involved in the care and use of animals for scientific purposes, and to develop or strengthen ethical review processes. As always, we will have a question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the top right panel of your screen, and I'll put them to Dave at the end. Details on how to access the on-demand recording of this webinar will be sent to you by email shortly. So now over to you, Dave, for the presentation.
Thank you very much, Adam, and also thank you to the audience for registering. So today I'm talking about the 12 R's framework, enabling high-quality humane research involving animals. Whilst I'm using animal research as the context, a lot of what I'm going to say with regards to the ethical issues and constructs are applicable to any discipline, whether it be animals, humans, the environment. A slight technical issue here. So if we think about research involving animals involved are any living species apart from humans. So we could go from the single cell protozoa through to water fleas, flies, fish, predominantly in Europe, rats and mice, but we could have farm animals or we could have wildlife, etc. Traditionally, we talk about these as laboratory animals, particularly in the global north, but the laboratory could be anywhere. It could be the animal facility, it could be the science lab, but it could equally well be the zoo, the wildlife park, the farm, etc. Purposes for which we use animals for scientific research, again, are varied. It could be understanding human and animal biology. It could be in the development of new medicines or therapies. It could be in safety testing and evaluation, higher education and training, and many other purposes. We've been undertaking animal research since 4 BC. The first law governing the use of animals in research was actually in the UK in 1876, so about 2,000 years later. Since then, in the 1960s, a few countries introduced laws governing animal experimentation, but still across the world, the majority of countries don't have laws or regulations governing the use of animals for scientific purposes. Same sort of picture with ethical review. In the UK, ethical review came in for animal studies in 2004, rest of Europe in 2013, but globally, many countries do not have ethical review or don't require ethical review of animal studies, although it is emerging in many countries. And we think about the ethics of animals, the use of animals in research, there's multiple perspectives and opinions reflected by the wide range of stakeholders. So, for example, we've got the public, we've got researchers, we've got the end users, those that are strongly opposed to animal research. As I said, there are strong views for and against, and people's perspectives sometimes change over time or with regard to circumstances. So we can think about it from 4 different ethical perspectives, and this was sort of published by the Nuffield Council for Bioethics. So there's the anything goes perspective, that the science is of benefit and therefore there's no longer any need to consider the ethics. The on balance justification, that there are benefits, although we're causing harms to the animals, and that the benefits to science, society, and human and animal health outweigh the harms to those animals. The moral dilemma, that we're going to harm somebody, whether it be animals, if we undertake these studies, or humans if we don't undertake these studies. Or the abolitionist, that there's no justification ethically for the use of animals research in research. Now, an individual's personal views are influenced by their values, society, culture, religion, regulation, time, and many other things. What I'm going to give you today is my perspective, coming up from a researcher who used to use animals for research, has now moved over to education and training. So my views have changed over time. Initially, when I started over 40 years ago, I didn't really think about the animal. Now my view is I would like to get rid of animal experiments in their entirety, recognizing this is probably not achievable within my lifetime, and therefore if we must use animals, we have to minimize the harms to them. So everybody's on a journey, and no individual or country has got that journey or has completed that journey. We're also on different journeys, that the end point might be different depending on your national laws, your own personal values, your culture, etc. So there's no way to say that the global north is right and the global south is wrong, or vice versa. But if you remember, though, is the principal actors are the research animals. The animals cannot give informed consent, that we have to be their voice, and that working with animals is a privilege and not a right. So if we are going to use animals, then we must minimize the harms. So the 5 freedoms were introduced for dairy cattle in 1965. So a freedom from hunger, a freedom from thirst, a freedom from pain and suffering. But recognizing it shouldn't actually be freedom, we should actually be providing these as a basic animal right. So Mellor changed the 5 freedoms to 5 provisions. So providing good nutrition, good environment, good health, appropriate behavior, positive mental experiences, with their associated animal welfare aims. So for example, good nutrition, minimizing thirst and hunger. Specifically in regard to research animals, then Russell and Birch in 1959 introduced the principles of humane experimental technique, or the 3 R's. And these really are the fundamentals of research, ethical principles underlying research involving animals. So replacement, so this is getting rid of the use of animals in research. This really should be our gold standard. So absolute replacement, where there's no animal involvement at all. So it could be using computer models or human cell or cultures. Absolute replacement, where there's some involvement of animal tissues or cells at one time. So animal cell cultures, animal cadavers, etc. If we have to use animals, then we should be minimizing their pain, suffering, or distress. So refinement. So appropriate environmental enrichment, pain, analgesics and anesthetics where appropriate. But not only think about minimizing pain, suffering, or distress during the experiment, but over their entire lifetime, from the moment they are born to the moment they die. And reducing the number of animals that we use. This now already been changed to optimization. So using too few animals, so that you can't statistically analyze your data, means those animals are wasted. So too few is just as bad as using too many, and therefore it's now optimization of animal numbers. So why should we apply the 3 R's? Society expects us to. In the UK, the government does a survey every year on public perceptions of animal studies. 60% agree with the use of animals for research on so-called, the conditional acceptors. Only if it's for medical rather than scientific research, there are no alternative to the use of animals, and they are minimizing harms to those experimental animals. Increasingly across the world, there's a legal obligation to apply the 3 R's. So it's implicit in UK legislation, it's explicit in the European Union's Animal Welfare Directive. and also a moral obligation of us as researchers. We shouldn't be invoking harms to or minimizing harms to other living creatures. So are the 3Rs globally applicable? I think yes, with caveats, and I'm going to explain that as I go forward. So if we think about the global uptake and applicability of the 3Rs, they were introduced in 1959, virtually unheard of until 1978, when a paper came out highlighting their lack of use or application. Where they've been implemented really depends on areas and roles. So particularly in drug safety and drug development. So, for example, the use of structure activity relationships to replace and databases to replace toxicology testing, or human cell skin culture to replace the Draize test. Again, in roles, more refinements and applications probably come from the animal welfare sector than scientists. There's also barriers and constraints. Industry in particular is spending a fortune on developing 3Rs methods. However, the scientific community is hesitant to take these up. There's a so-called 3Rs valley of death. And there are many reasons for this. So if you've been doing animal studies using a particular model for 30 years, are you going to suddenly say that the data is irrelevant, use the wrong model and therefore switch to a totally different one? It's not in human nature. But also we know a lot about the anatomy, physiology, pharmacology of a rat or a mouse. We know very little about the same in a worm or a fly. And therefore we have to get all this background information before we can move on and advance the science and build on those things. But also constraints as we look across the world. If we take Africa, then laboratory plastics, basic consumables, and chemicals and basic kit is very expensive to import. Therefore, in Africa, many studies that in the global north we might use non-animal experimental approaches to address. In Africa, in other resource-constrained countries and regions, animals are used. If we look at legislation, there are some countries where the 3Rs is in the legislation but not in practice. So in India, have to, the 3Rs are in national legislation, but if you look at their government or national animal ethics form, the 3Rs aren't mentioned. In the US, you only have to think about the 3Rs, you don't actually have to apply them. But as I said, it is very much a global journey, and I don't think any country in the world has got it right yet. And that journey is going to differ depending on our individual values and cultures. And that's no more evident than working with colleagues in Africa, some who are on this call, where we created guidelines for ethics committees in Africa. Couldn't, translating global north guidelines and procedures wouldn't work for Africa. Different culture, different legislation, etc. So there's very much the need to create something that is relevant to your particular situation. But also, when we talk about animal experimentation, we have to think beyond the ethics of animal research. There are many other ethical issues and constructs that are also come into play. And these include, are the studies ethical? And again, these apply not only to animal research, but also to all other areas of scientific research. So are your studies ethical? Is there any alternative to the use of animals, or for using humans, the use of humans, or environmental studies if you're doing studies in environment? Is what you're doing acceptable to society? We're doing science in society's name. We shouldn't be doing things that are not acceptable to our societies. Is what you are doing permitted under national legislation or regulations? Again, these vary considerably across the world. What are your personal values? So again, taking animal experiments as a content in context, then I have colleagues that will work with rats and mice, but not higher order species. I have other colleagues that will work with dogs, but not monkeys, or vice versa. Also, the types of studies that people were prepared to do. Some will only work with freely moving animals where there's minimal, if not no, pain, suffering, or distress. Others are quite happy to do more invasive studies that do cause pain, suffering, distress, and potentially death. Your research culture and integrity. This is a big thing at the moment across the UK. You know, there are scientists that act without integrity, so there is fabricated data and falsified data out there. Do your studies minimize the harms to your experimental subjects, so animals, humans, or environment? And are you maximizing the benefits to individuals, to science, and to society? Is your research reproducible, reliable, and translatable? So there's an awful lot here to take on board, and particularly for those new to considering ethics, it can be quite difficult. And therefore, we created the 12 R's framework, which is a visual representation of all these different ethical issues to support colleagues in implementing them in their own scientific endeavours. I have to here acknowledge Professor Tian Brink from Northwest University in South Africa. Tian is a good friend of mine, and he was the brainchild of this 12 R's framework. It was his concept originally. I then worked with him to modify it and adapt it. So recognizing Tian's input to what I'm talking about today. So if we take it, we can really split it into three groupings of R's. So we have the animal welfare R's, which I've just talked about: replacement, reduction, and refinement. We have the social value R's of responsibility, respect, regulation, and the scientific integrity R's of reproducibility, relevance, and transferability. So we've talked about the animal welfare R's, now talking about the social value R's. So there's responsibility there towards the animals, to researchers, trust from society, the environment, integrity. There's respect for animal dignity, so-called culture of care, 5 freedoms, or as they now are, the 5 provisions. In the UK, we buy our animals off the animal facility, they become our own. In other parts of the world, you rent them off the animal owner, so the farmer, the game park owner, etc. So it's respecting the owner's rights as well as the animals. Regulation, so legislation, regulations, directives, standards, etc. Take the scientific integrity R's. Then we have relevance, so we just study justified, to add value to science, real and likely benefits for animals, humans, society, etc. Transferability, is it transferable to other animal species, including to humans? And reproducibility, is the study design appropriate? Is it statistically valid? Are you transparent in reporting and sharing of data and information? So those are the 3 groupings of R's, and you can see there's also overlap between the different circles. So reliability, reckoning, and righteousness. Built into this are other ethical principles, so prioritarianism, virtue ethics, principalism, care ethics, deontology, and utilitarianism. But overriding it, where interlinking all 3 sectors, is a harm-benefit analysis. Now, this brings together as a visual representation all those different ethical constructs I've just talked about. It can be simplified and applicable across all areas of research. So effectively, 3 pillars. Again, to all disciplines: social values and scientific integrity, and then the welfare pillar, which could be animal welfare, environmental welfare, human welfare, etc. Linking all 3 is integrity and honesty, culminating in a harm-benefit analysis. Once we've just done this, then it's, we haven't just filled our form in, got the approval, tick the box, and that's it. It's a continuous monitoring, reporting, consultation, improvement, etc., as we go through our scientific research and beyond. It involves all stakeholders and role players, so researcher, student, ethics committee, professionals, institution, etc. And so this is in practice. You as the investigator come up with a concept, you ensure that you have appropriate education and training, you consult with experts, statisticians, animal welfare experts in your planning, you then seek ethical approval, they might come out with recommendations for you to go back to replan, etc. Or they might give you permission, in which case you execute it, but you monitor animal welfare and other things as you go through. You might change things if animal welfare is going downhill. You report it to the scientific community, you conclude it, and then you keep your records and use that information in subsequent studies. So it's a dynamic consideration and active application of 12 R's pre, during, and post-study, and involving these different communities. So it is very much a work in progress across the world. And again, if we just talk about each of those three circles in turn, if we take, and it's continually evolving and developing. So if we take the animal welfare R's, globally we brought in additional R's beyond the original three. So in India, there's rehabilitation. In Africa, there's respect and responsibility. Our gold standard should be ultimately replacing the use of animals in research. That is a clear direction for the European Union Animal Welfare Directive. There's an increased development of non-animal models as alternatives to the use of animals. And just recently in the US, the Federal Drug Administration permitted studies to go into clinical trials using non-animal safety tests rather than the normal 2 animal species. Collectively, we need to broaden the loci of change beyond the sort of safety pharmacology, toxicology sector, beyond the animal welfare colleagues, into the broader areas of research and also more into the scientific community. Looking at ethical review applications as a member of an ethics board, you often see things that are, under refinement, that they're providing appropriate housing, they're providing food and water, etc. These aren't refinements. Those are things that we're actually legally required to do. What would be a true refinement would be, instead of just giving a dose of anesthetics, you actually gave it a dose, then you looked at the impact on the animal and did a welfare assessment and decided whether it needed more analgesic on top of that. I think there's a need across the research and welfare community to understand that these refinements are summative, so that more than the sum of the individual parts. So by more appropriate handling, by environmental enrichment, diet, etc., all of these build together and enhance the welfare of the animal. There is a need for greater global uptake, and again, as appropriate. So as I said before, animals are used in Africa in situations that they wouldn't be used in the global north because of resource constraints. But I think it's critical that as we increase our understanding of animal welfare, of the 3 R's, etc., and through our own research around animal welfare interventions, that we share this learning across the global community so we can all apply them. Scientific integrity R's, reproducibility, reliability, and translatability of science across the board is poor. So this survey in Nature a few years ago showed that 70% of scientists couldn't reproduce somebody else's research and 60% couldn't reproduce their own research. Animal research is even worse. Up to 95% of animal research is neither reliable nor reproducible. Why are we doing it if we can't believe that data? This is critical to the way that science actually develops. So you can see science developing as a glowing tree. We build on existing knowledge, we add more branches, thicken those branches out, etc. If we can't believe these earlier studies, then we're really starting from scratch every time. We're not advancing science as we should. Now, the decrease in reliability and translatability of animal studies, animal welfare has a huge impact on this. So animals are living beings. What impacts on us will also impact on them. So what's inside the cage, what we feed them, gut microbiome, how the mother interacts with pups, how we handle them, the time of day of studies, the background strain for genetically altered animals, the gender or sex of the animal. All these things will impact on our data and might even change our data. And therefore, we need, as a community, to report this information. So this is where Arrive and Prepare come in. Arrive is the Animal Research Reporting in Vivo Experiments guidelines. It's a checklist of 13 things that you should put in your paper. So it's things like strain, species, number of experimental animals, statistical technique approach, etc. So that others can replicate your studies because you know exactly what you've done. Often you can report these as supplementary information. Prepare is planning, researching experimental procedures on animals, recommendations for excellence. It's not a checklist, but an aid memoir of things you should do before you start your study. So making sure that everybody has appropriate education and training, is competent, have you consulted with your animal welfare colleagues, etc., plus all the Arrive 13. So Arrive is sort of post-study, but you really need to gather this information as you go through so you have it there when you need to report it. And by following both Prepare and Arrive, it will make sure that your data is more reproducible. Increased use of systematic reviews. So these are where you look at all the published information that specifically addresses your research question and then do a meta-analysis of the data. So you can get true things rather than trends. It's an approach that is used extensively in clinical trials to bring together data from multiple clinical trials. It's now expanding into animal research. In fact, in Switzerland, it's a legal requirement that you must do a systematic review before you can undertake a study involving animals. And increasingly across the sector, there's a focus on experimental design. Many of us weren't trained, haven't had any training in experimental design. There's an increased number of courses coming on stream to actually give us that required training, which will increase the reproducibility and reliability of our research. And social values. Critical this is a culture of care. So a culture of care is a commitment for everybody in the institution involved in the care and use of animals, from the head of the institution right down to the animal caretaker, to improving animal welfare and the application of the 3 R's and 5 provisions, to enhancing scientific quality, to enhance the care of staff involved. Use of animals is very emotive. So, for example, if you've got an animal caretaker who you're expecting to cull animals at the same time every week, is he using the most appropriate technique that is less emotional for him or her? Do you give him the choice on the time he does it? Do you share the job around individuals? Transparency for all stakeholders and a compassion and respect for humans and animals. Research culture, I've talked about. It's enabling high quality science, through equality, diversity, inclusion, through enhancing research integrity. In the EU, there's a requirement for retrospective review. So this is reviewing a study on completion and sharing on, reflecting on the animal welfare implications, any improvements that could be made, and sharing that knowledge. Again, it should be shared more widely. As I talked about earlier, there's increasing regulatory change. More and more countries are introducing laws that govern animal experimentation. And there's also those that have them are strengthening them. So in the UK, for example, consultation at the moment about including decapods into our legislation. But also thinking about learning from the global south. The global north hasn't got it right. We all have issues, as I've highlighted today. And there's an awful lot we can learn from the global south, particularly from rural communities. There's an impression, particularly in those communities, that they should transpose global north perspective, procedures, etc., to global south. As I've hopefully shared, it won't work. You have to develop things that work for your own particular communities, cultures, etc., but also learn from different parts of the world, sharing the good practice. So when I've given workshops on animal welfare in Africa and other parts of the world, I often get the comment, white man's view of animals. They're right. I'm giving you my perspective. I have given you my perspective so far. Your views might be different from mine depending on your culture, values, etc., religion. African Union has an animal welfare strategy that says that in Africa, where animals are treated as sentient beings, as a leading continent in the implementation of good animal welfare practices for competitive and sustainable animal resource sector. They're the only continent that I know of in the world that actually has that commitment. Yes, in the European Union, we have our Animal Welfare Directive, but animal welfare under that directive is constrained by two other EU principles. So the free movement of labour and the not bringing in procedures or regulations that confer an economic advantage. So when the directive came in 2010, then the UK was allowed to keep its higher animal welfare standards because those were in place before the directive came in. Other countries had to stick to the agreed minimum. Italy wanted to enhance its animal welfare standards after the directive came in, but wasn't because it contravened those two other EU principles. Thinking about again from Africa perspective, the One Health triad, that you can't think about animal health in isolation from human health, in an environmental health, that you really have to think about it as a single entity, that one will impact on the other. And if we are trying to solve some of the complex issues in Africa, we can't just focus on environment, we can't just focus on animals, we can't focus on humans. We have to think about the three in unison. And an outcome of that is how African rural communities think about animals. So responsibility. We're thinking about my global north perspective, when we think about minimizing harms to animals, so refinement, we think about the individual animal. If you take that, and if we think about meat production, we just get rid of those animals that aren't bulking out, etc., and cull them early. We take Africa and sub-Saharan Africa, the camel herder. His animals are his source of meat and milk. They're a source of income. The hides might provide shelter, and they're his bank. He can't just think about the individual animal. He has to think about the whole herd. He has to have the animal that is the good breeder, the animal that is the leader, the animal that has the maternal instincts, the animal that has able to best survive in a hybrid conditions. So but what I think about the whole collective, then he has a herd that will survive. Think about respect and the Rift Valley. Then individual families or clans within the Rift Valley adopt an individual animal or species as their totem, their good luck symbol. They provide it with shelter, they provide it with food, they protect it from predators. You don't harm another animal because you're harming another clan's or family group's totem. So I think there's an awful lot, just two examples there, but I think there's an awful lot the global north can learn from the global south. So just bringing it all together, then 12 R's is driving forward change. It's applicable across many different areas, not only just to animal research. It brings together in a visual representation many separate ethical issues and initiatives. It's synergistic, that you can work, you need to improve both the animal welfare and the scientific integrity R's or social value R's at the same time. We need to raise awareness of the interrelationships and therefore to have greater impact on our science, more high impact science, more reproducible science, that they are applicable globally and across all disciplines and areas of research, and a recognition that the direction of travel will change depending on different cultures, different regions, etc. That we're all going down a journey, but our endpoints may differ or change as we go down that journey, both individually and as countries. Thank you very much for your time. Now open to questions.
Well, thanks, Dave. That was a very interesting presentation. Thank you for sharing that with us. We have a few questions from the audience. If anyone else has a question, please feel free to post it into the questions box, which appears on the top right panel of your screen. Okay, so Dave, the first question, I know you touched on this, but are Western ethical constructs and issues applicable globally?
I think, as I said, yes and no. So there are ones that we can apply, but then modify depending on different contexts and cultures. So the 3 R's, as I said, are applicable, but refinement is not always possible. Replacement is not always possible. Refinement may vary depending on what you're allowed to do in your own particular country.
Mm-hmm. Okay, thank you. And the next question, which R takes precedence?
For me, it is replacement, getting rid of the use of animals, at all. Then it moves on to, for me, it is refinement. That if you think about from the animal's perspective, then we should be minimizing, sharing out the suffering, rather than having an individual animal that has multiple, lots of suffering. Then we should be sharing it out across the animal community, but also that little things, so how we house them, how we handle them, etc., can have massive impact on the animal because they all add up. So by doing very small things, we can have, from an animal's perspective, we can have a massive impact on it. So for me, the pecking order is replacement, refinement, and then reduction.
Mm-hmm. Okay, thank you. Okay, and so I think there's another question. Getting ethical approval is a very bureaucratic exercise with limited benefits. Why should we bother?
Then for all things, so in many countries, it's a legal requirement. If you want your studies to be published, then you will require ethical approval. But I think the overriding thing is morally, we've got to be doing best for the animal. By sending it to your application to an ethics committee, it's a new pair of eyes. They will pick out things that you haven't missed, that you've missed. They will give you advice based on their knowledge, etc. Your study is going to be much better. You're going to minimize harms to the animals. Your science is going to be far more impactful.
Okay, thank you. All right. Next question here. What would you recommend when doing research on production animals in an industry commercial setting when the industry norms are lower standards than what is expected for research animals? That one's from Margaret.
Yeah, so thank you, Margaret. Really good question. So there are two different drivers there. Industry is very much, how can we maximize profits? But on the other hand, as research animals, if you have the lower welfare standards, it's highly likely that your data is not going to be reproducible and reliable. So therefore, I would go for the higher standards.
Okay, thank you. And another question, how do you prioritize the 3 Rs for a country with limited resources? Do you have a suggestion?
I recognise that animals are used in resource-deprived countries because there's no alternative. They don't have the non-cellular based approaches. I think if you can use them, can get the resource to do it, then it's ideally yes. The alternative would be to collaborate with people in more resource-rich countries. So, are there parts of the bigger study that you could do best, and parts that they could do best based on resources and approaches and availability of equipment? It is a difficult one when it comes down to the number of animals. So, in some countries, then your studies are underpowered because you don't have the resource to have the suitable number of animals that you actually need. This is a difficult one. If we said don't do it at all, then it would kill research in that particular environment. But your data is not statistically analyzable. So I think for me in that circumstances, then it's report it, but report it as a trend, not as a fact. That this is what you've seen in the few animals that you could actually undertake it, rather than taking it as a scientific given.
Okay, thank you. So the next question from Pradeep, it says, thanks, Dave, for a wonderful presentation. Where and how do you think tech advances in structural and cell biology integrated with machine learning could help us mitigate animal testing? It's an interesting question.
Lovely question. I actually saw a brilliant example of this the other week in Karolinska Institute in Sweden. So what they were doing was looking at lipids within the embryo of the liver of an embryo of a zebrafish. And so we're into the sort of, you've got to use a microscopic thing. And what they were doing was actually using machine learning to identify the individual liver cells within the embryo, but also the lipid constituents within it. And then it became a high throughput screen because they could, a 96 well plate, an embryo in each one. So there's huge potential out there. And I think we need to talk to all different disciplines, the computer specialists, the analytical chemists, the animal biologists, the invertebrate biologists, etc. And there is huge, I was amazed by some of the things they were doing using AI in sort of invertebrate models.
Yeah. Okay, thanks, Dave. Next question. How do you ensure that individuals understand that animal welfare and good science are interconnected instead of believing that one is more important than the other?
Thank you. I think they are equally as important, and explicitly interrelated. If you have poor animal welfare, then your science is going to be of low impact. And that really it is educating researchers that they need to talk to the animal welfare experts and the design and execution of their studies, and that even small changes can have a major impact on the welfare of the animal and the reproducibility of the science. So I think it's very much an education thing that everybody, all researchers want high impact science, but if you don't get the animal welfare right, your science won't be as impactful as it could be. But also, there are times where animal welfare has to be compromised in undertaking the science. So, for example, if you are culling an animal because you want the brain tissue, you might use a less humane approach to cull that animal, because if you didn't, if you used a more humane approach, it would ruin your tissue, and therefore the animal will actually be wasted. So I think it is an understanding of both the scientific issues and the animal welfare issues, which is an education thing, but also then weighing up the balances. How is it going to impact my science by enhancing welfare, and or how can I enhance welfare to impact science? And doing that sort of, for want of a better word, harm benefit analysis at every stage.
Mhm. Okay. Right, Dave, I think we've got time for a few more. This one from Claude. I'll try and get it right. So, most research ethics committees in Uganda are for human research participants with only one accredited IACUC at Makerere University. What is your advice on using research ethics committees for human participant research to approve research involving experimental animals?
I understand that, and I know actually it's changing in Uganda. You brought in your animal ethic guidelines, and they've actually got a course that I'm participating in on the 8th of February. But I think it's the same even with research ethics committees. You've got to have the individuals on there who are capable of doing the ethical review of the science. So in a human committee, you've got to have those, and it depends on what science goes into that human committee. If it's sort of sociological research, you've got to have the sociological expertise. If it's physiological research, you've got to have those who are used to doing physiological studies. And so if you are getting ethical review of animal studies through your normal research ethics committee, you've got to have what you'd participants on there. So somebody who actually understands the science, and an animal welfare expert on there. So it's making sure that your committee has the appropriate representation and expertise on it to appropriately review your particular study.
Okay. All right. I think time for one more question. It seems like quite a broad question that you've covered, but what is the relation to the 12 R's and the 3 R's, and how do we go about this in vaccine production?
So the 3 R's, replacement, refinement, reduction, are a subset of the 12 R's. So there are an additional 9 R's in addition to the 3 R's of animal experimentation, and they're all interrelated. With regard to vaccine production, then there will be the research involving animals to develop the vaccines themselves, which will have the animal welfare R's involved. The other 9 R's, the social integrity, translatability, and the social scientific integrity and the social value R's, will come into that as well. When you come into the vaccine production, and you might be using animals to produce the vaccine as sort of bioreactors, then again, all 12 R's will come into it. The animal welfare R's, particularly looking at the welfare of the animal, and then the other 9 R's when it comes to, as I said, translatability, relevance, etc., of your scientific information.
Mm. Okay. All right. I think one final question, Dave, because I think this is quite important to touch on again. So this is from Farida. Do you think the 12 R's framework is really applicable in the same way globally between North and South?
The basic framework is, I think the different R's are translatable to a different extent. We might put more focus on one than another, depending on our culture, our constraints, etc., our values. Um, and but and but yes, I think they are. But we will, we're not all going to maximize wherever you are in the world. We're not all going to maximize all 12. There are different ones that are more applicable, the ones that are more implementable, etc., across the world. And as I've said throughout, it's a journey.
Mm.
We're all at different stages of that journey. We might focus on different ones as we go forward. We might have different endpoints at the end, but we're all on that journey. And there is nobody anywhere in the world has finished that journey, and the journey is different for different communities.
Great. Okay, super. Well, Dave, I think we can wrap up there. That brings us to the end of today's webinar. Thanks again, Dave. That was an excellent presentation and some great questions too. So thank you for that discussion. And finally, thanks to the audience for taking the time to attend and listen in. So until next time, good luck in your research and goodbye from all of us at Bite Size Bio. Thank you.
And my thank you to all as well.